I'm really delighted to welcome Professor Sir John Byrne, Professor of Clinical Genetics at Newcastle University and Honorary Consultant Clinical Geneticist and former Chair of Newcastle Hospitals. Today he's going to talk to us in advance of Lynch Syndrome Day, which is actually on Thursday, to tell us a bit more about the condition that he's certainly spent over 30 years researching. So please welcome Sir John Byrne, and I'm going to hand over to him.
Thank you very much, Nikki, and it's a pleasure to be with you today. So I'm going to rattle through this story. As Nikki says, it's more than 30 years old, so there's quite a lot to tell, but it has really evolved enormously in the last few years. So I should just say, I haven't got a declaration slide, but I'll be talking a little bit about the MSI Plus assay we've developed later in the talk, which is relevant specifically to Yorkshire, and I am named on the university patents for that. So I'm going to cover a number of things. First of all, many of you, especially people, older clinicians, will remember the term HNPCC, and I'm going to explain why we switched away from that, what the mismatch repair genes are and how they lead to cancer and how we can prevent that. And then my next section will be a little bit about colonoscopy and some rather surprising data on that. And then how we treat it and what other cancers should we watch out for and how do we test for them. So the term HNPCC was hereditary non-polyposis colon cancer because back in the late 80s, we'd found the gene for familial adenomatous polyposis. And these were young people like Simon, that was his colon, taken out 3 days before that picture was taken, with thousands of polyps in their colon leading to cancer almost inevitably by the age of 40 to 45. And fresh from the success of finding the APC gene and being able to track it in families, we still couldn't find mutations very easily, but we were able to tell Simon's sister that she had the other copy of the gene and was in the clear. And we learned quite quickly that this was a very important gene. When we look at the crypts in your colon, down at the bottom there are about half a dozen stem cells, and the APC gene is critical to actually keep the system working well. It's a very big gene that it stops beta-catenin, which causes cell proliferation. It helps to segregate your chromosomes properly, and it manages cell migration up the crypt to the surface. So if you lose APC, you lose all sorts of things, and all of those functions lead to cancer. But we were interested at the time of finding this gene in families where there was clearly colon cancer running in the family, but we couldn't see lots of polyps. And so to bring them together for a linkage study, they were called hereditary non-polyposis colon cancer. And the name sort of stuck, and it was a rather silly name, but it was used for quite a long time. This was one such family, which was actually written up in 1971 by a GP called Ted Naggs, who's in the picture there outside our medical school down in Durham. And when he wrote his paper in 1971 with Dr. Dunstan at Durham, they'd had 45 bowel cancers in one family. And remarkably, people back then said, oh, it doesn't run in families, it's just coincidence. And clearly that wasn't the case. And so Tim Bishop in Leeds, my partner in crime for all these years, said, let's try and map this. And indeed, we started mapping a couple of families. When we were contacted by the yeast geneticists in Boston, and that was Richard Kolodner's group. And basically, we then collaborated with them, and they identified first MSH2 and then MLH1. This family was an MLH1 family. And Brian there at the front, who's at the back of the picture there, he didn't even know he was a member of this family. And when we got him in for a colonoscopy, he had a huge cancer in his rectum, which was successfully resected. So this was a very sort of early payback on mapping these genes. And there was a huge flurry of activity back in the 93 to 94, 95, following this paper from Richard Kolodner's group, was in Cell in December 93. And this family from Northumberland was used in that study. And indeed, they found a mutation which actually wasn't the pathogenic variant, but it allowed us to track the gene in this family. And so we think this is the first family in the world to get a diagnostic test off the back of discovery of these genes. So Ted there had bowel cancer 3 times and he had 9 skin cancers. And we thought this was another condition called Muir-Torre syndrome, which turned out to be a subtype of Lynch syndrome where you get skin cancers as well. And his sister had endometrial cancer, which is the classic other cancer that we see most commonly in this condition. And the very fact that you see both skin and endometrial cancers explains why HNPCC was a bit of a stupid name, because it isn't just colon cancer. There's a whole range of cancers in this condition. We now track about 8,500 people with Lynch syndrome across Europe in the prospective Lynch syndrome database. And we know that from 25 onwards, there's a steady increase in cancer, such that by the age of 75, 80% of carriers of this gene, the MSH2 gene, will have developed a cancer in either the colon, endometrium, skin, kidney, small bowel, etc. So there's lots of places they could get cancer. So this is a really important condition. And also it became apparent that this was, in fact, a very common condition, about 3 per 1,000 of the population. So we think there are about 150,000 people in Britain with this condition. The box there is around a young man called Stanley, and he actually led us to this family because he had familial adenomatous polyposis. And we thought that this family was connected to that. It turned out he'd inherited it from his dad, who interestingly was sent out to Christmas Island to clear up after the nuclear tests. And a double strand break in his sperm was what gave Stanley a deletion of the APC gene. And then quite separately, he inherited Lynch syndrome from his mother. And he got colon cancer 3 times before he was 30. So that's Henry Lynch, who sadly died in 2019, an old friend. We worked together for many years. And Henry was a very larger-than-life former heavyweight boxer, Irish Catholic, who trekked the world telling people that this condition existed. So we named it after him, even though he wasn't the first to describe it, because it was a better catch-all for this fascinating condition. This is my friend Chris Heinen in the University of Connecticut, and he has been working on the biology of this system for a long time. And basically the reason we made such rapid progress was that once the first gene was found, and it was a hunch from the yeast geneticist, because the thing with these cancers is they get thousands of spelling mistakes. And so they figured it was probably a problem with the spell checker, which turned out to be true. What's remarkable is that yeast uses essentially the same genes as humans to do this spell checking. And in fact, every cellular organism uses this bunch of genes to spell check its DNA when it's copying. Chris made this little animation about how it works. And so MSH2 is the pink one there, and it partners with MSH6. And a little later on, you'll see MLH1 come in with its partner, PMS2. There are other genes in the set, but they're the 4 that matter. So basically, when there's a mismatch where the DNA molecule doesn't form, these clamps load onto the strand and catch up with the polymerase until they've filled it up to the point where the error is. And then the MLH1 and PMS2 activate effectively that clamp system. That then triggers XO1 to strip the strand back to where it was wrong and run the polymerase again so that you actually get the correct letter. So there's the 4 genes, MSH2, MSH6, MLH1, PMS2. EpCAM is actually upstream of MSH2, and it's got the promoter for MSH2 sitting within it. So if you delete EpCAM, you'll take out MSH2. And the other thing you can get in MLH1 particularly is you can silence it with methylation. And most sporadic cases of mismatch repair are due to silencing of MLH1. And occasionally people get that happening in their germline or very early in development. So they look like they've got Lynch syndrome, but their germline DNA is okay. It's just that everything in their body is running with a faulty copy of MLH1 because of that methylation process. So that's the biology in a nutshell. And what that leads to is something very important, which are frame shift peptides. So the letters here describe the amino acids in a very important gene called TGF-beta receptor 2. And TGF-beta receptor 2 has 10 letter A's in the middle of it, and that's called a microsatellite. It's an old term for bits of DNA that run off the top of the gel. And it's these bits of DNA that are particularly prone to copy incorrectly when you're replicating your DNA. And so if you drop from 10 to 9, then first of all, you've obviously changed the sequence of the DNA, but crucially, you've caused a frame shift, because all the genes are read in sets of 3, so everything moves back 1, so that the amino acids from that point on will be different. You come to a premature stop codon. If you go back to 8, you get a different premature stop codon, and you get these so-called frameshift peptides that leak out of the cell, and they're highly immunogenic. So these tumors get seen by the immune system, which I'll come back to later. But actually, crucially, for several years now, colleagues have been trying to develop vaccines based on this. And there is actually one called from Nuscom, which has a couple of hundred of these frameshift peptides loaded with viruses. And they've demonstrated the safety of those viruses in patients with Lynch syndrome at Sloan Kettering. The 2 viruses we used to develop the Ebola vaccine, and it's essentially the same principle. Instead of putting the Ebola vaccine DNA into the viruses, you put these frameshift peptides. The idea is you immunize people against cancer in this condition. We're not quite sure whether it's going to fly, but it's causing great excitement at the moment. So TGF-beta receptor 2 is down on the bottom line there, and it's one of the genes that you see in the progression of mismatch repair gene defect cancers. And this is a different pathway to the APC gene, where you get a different collection of mutations and what's called chromosomal instability. And that leads to what we regard as common or garden bowel cancer. Most cases will follow that sort of route. So a mismatch repair deficiency, whether sporadic or inherited, produces a different collection of mutations because of the spelling mistakes in these genes, and it produces a different molecular profile. And about 1 in 6 of all bowel cancers, which occur in about 37,000 people a year, about 1 in 6 of them will have a breakdown of mismatch repair. Most of them are sporadic. And of the sporadic ones, about half of them will have a mutation in this gene, the BRAF gene. Now, this is a driver mutation. It's not one that you see in Lynch syndrome, hardly ever. So basically, we test for that as well, because if you've got a BRAF mutation and an MSI profile, you're probably sporadic and we don't need to pursue it. And I'll come back to that later. So there's TGF-beta receptor 2 I described earlier. So in a way, Lynch syndrome patients are a kind of genetic model for the people who've got this type of bowel cancer or this mechanism for developing cancer. BRAF is part of a pathway called the RAS-MAPK pathway, and it's very important to oncologists because one of the drugs we use to treat bowel cancer and other cancers is an EGFR receptor inhibitor. And if you block EGFR, then you block cell proliferation. However, if you've got the BRAF mutation, which is a point mutation, then you end up with an activated BRAF, regardless of what the EGFR receptor is telling it. So if you've got that, then giving patients the drugs to block EGFR won't work. So it's really quite important to know about that when you're deciding how to treat a bowel cancer. So I got into hereditary cancer quite late. I was a disc morphologist through the 1980s, but having got involved in finding the FAP gene, we were looking for families in our region to set up a register, and this was a family we visited at Monk Wearmouth. And Jonathan in the background there was 12 years old. He just had a colonoscopy and he was clear. His mother had thousands of polyps. That wasn't her colon, but it looked like that. And she'd had a pan-proctocolectomy. But what was striking was that both mother and son had osteomas on their forehead, little bumps on their forehead, which are a classic extracolonic feature of FAP, or familial adenomatous polyposis. So I knew he was going to get it, but he didn't have any polyps yet. And that made me think, wouldn't it be great if we could do something to slow down the progression of the disease so he didn't have to have a colectomy at the age of 18? And that triggered the idea of targeted trials. Could we do genetically targeted trials, not only for the people who've got the genetic condition, but because they are a model, if you like, for the general population? And by chance, Monk Wearmouth is right next to a pit, or was a pit, and I went round to see them and managed to get a picture of their canary, or one of their canaries. So this is a canary that the miners used to take down the pits with them because they would fall off their perch when there was gas in the air, and they would rush them out and give them mouth-to-beak resuscitation and save all the miners from death. And I figured that, in a sense, Jonathan was a sort of canary down the mine for us. So that if we could work out a way of stopping him getting cancer, then that would be of relevance to the general population. Just as we started a trial, which became CAP1, in about 130 people with FAP, we were involved in the discovery of the mismatch repair genes. And so we started our CAP1 trial in 1993, but then along came Ted and his family, And this was obviously a much better target population because they got cancer later, but they didn't have thousands of polyps. It's quite hard to know whether you've made a difference when you've got so many polyps to count. So we started CAP2, which turned out to be the much more powerful study off the back of this concerted action from European Union, which then became our cancer prevention program. So scroll forward 30 years, and this is the paper that we published in 2020, which won the ASCO prize for the best prevention paper of the year. And this was basically following up 1,000 people with Lynch syndrome in 43 centres in 16 countries, and we gave them 2 aspirins a day or 2 placebo tablets. And remarkably, it worked. And so if you look down at the left-hand side here, we treated people for 2 to 4 years with either 2 aspirins or 2 placebo tablets, and then followed them for 10 years without anybody knowing what they'd been on. And then the subsequent 10 years in the UK and in Finland, we used our registry data to see what happened next. And the impact was fairly glaringly obvious, as you can see, that the people who were on the aspirin had less cancers or less bowel cancers. And so for every 25 people we gave 2 aspirins to a day, we prevented a bowel cancer, which is cheap as chips and clearly very attractive. And interestingly, there was no significant difference in side effects between these 2 populations because younger people tend not to get side effects from aspirin. So if you look at the incident rate ratio there, which is in bold, that's taking account of all their bowel cancers rather than just their first cancer, which you get from hazard ratio. So the incident rate ratio shows that there was a highly significant reduction, 50% reduction in bowel cancer. And on the back of that, NICE actually then recommended that everyone with Lynch syndrome should be offered aspirin as a preventive agent. Now, that's not yet in the British National Formulary, and partly because we weren't sure what the best dose was going to be. We'd already decided to set up another trial, which will test the doses. But it wasn't just our study that showed that this worked. In fact, we had evidence going back to 1990 that people who took aspirin seemed to get less bowel cancer. Nancy Cook and colleagues led a parallel study to ours in a general population of about 18,000 women, where they gave them a baby aspirin every other day, alternate with vitamin E. And at 10 years, they'd seen no effect and they stopped the trial. But then because of our results, and some that I'll show you in a minute, they went back to follow up what happened next in their trial and published this paper in 2013. And you can see that after they finished the trial in 10 years, there was this divergence, so that it wasn't as dramatic as ours, but they saw about a 20% reduction in bowel cancer for the people who'd been on the aspirin, or to be precise, there was a 20% difference. There was a slight worry here that it looked like the placebo sort of took off. And one anxiety was that people started taking aspirin when they were told they'd been on placebo, and that maybe there was a little surge of activity there. But whatever, the point is that it was different, but it wasn't as dramatic as we found. The other equally dramatic result came from Peter Rothwell's study. So Peter came from Oxford, he's a stroke doctor, and he went back to the original trial of aspirin as a prevention of heart attacks, and tracked down, along with all the other aspirin trials of the 1980s, all the people who'd been given aspirin or placebo to see if it prevented heart attacks or strokes. And then they looked them up to see what happened next. And as you can see, that from about 5 years out, there was a very obvious divergence, so that from 5 years on, the people who had been given aspirin in that original trial ended up with less cancer than the people who had been given controls. Now, the other thing, just to remind you, we stopped giving the aspirin to our Lynch patients after 4 years, and we were still seeing an impact on their cancer rate at 15 years. So basically, aspirin is in some way helping the body to kill off the cells that are going to grow up to be a cancer in the future. And we're still working on how that what the mechanism of that is. So the other thing, so basically there were 21% fewer deaths from cancer in the people who were given aspirin. The other thing we discovered, we knew that obesity was a risk factor for cancer. So we went back and did a secondary analysis on our CAP2 data with Tim Bishop and the team in Leeds leading on this. And we said, what was the difference that obesity made? And it was quite dramatic. So people who were overweight were 2.5 times as likely to get a bowel cancer. So for every unit of BMI, there's a 7% increase in cancer. What was also remarkable was that that was most obvious in the placebo group, where the risk was 2.75, and it was non-significant in the people who were in the aspirin group. So it seemed that the aspirin might be actually negating that pro-inflammatory effect, perhaps, of the obesity. So it might be helping particularly people who are overweight to reduce their cancer risk. For the clinicians and the nurses on the call, just to take note of this. So people who've got Lynch syndrome, we really need to work on their obesity problem, which, as you know, affects 30 to 40% of the adult population now, because it really puts them at a very much higher risk. The other question, of course, was why is it not in the British National Formulary? Well, because we don't quite know whether a little dose is as good as a big dose. So we set up another study called CAP3. And here is Nick on the top right there. I went to see at the weekend at his workshop. He makes furniture in Newcastle. He's now 10 years out on aspirin and still going strong and has no side effects. And Robin at the bottom there, I saw last week. He was our last patient, last visit for the CAP3 trial. So in total, we recruited 1,879 people in 5 countries to take one of these 3 doses, blind for 2 years, so we could look at side effects. Then we did open label for another 3 years to see the maximum impact, because we're only counting cancers, so it doesn't matter that they know what dose they're on. For the majority of them, they were agreed to stay on whatever dose they'd been prescribed, almost all of them, in fact. And so what we'll actually, I should just say that we've got last patient, last visit from Melbourne in June, and then we'll break the code on this. And before next summer, we've got to analyze all the data. We'll see whether we see the same benefit from a small dose as a large dose. And obviously, if a small dose works in Lynch syndrome, we can recommend that. I mentioned that there was something strange about the Nancy Cook study. Well, there's also something worrying about the use of aspirin in old people. And that is that people who take aspirin when they're old get lots of side effects. And there's also a suggestion from a study in Australia that maybe the aspirin wasn't as effective in that group. And a similar study many years ago in California suggested the same thing. And it might be that the colon is actually changing. So this is Laura Greaves, my partner in Newcastle, who works in mitochondrial genetics with her team. And back when she was a student, she did a paper, this picture in the center there, looking at mitochondrial function in old bowels. So by the time you get to my age, around about 10% of the bowel crypts have got mutated mitochondria. And that means that the brown stain here is not formed. You get a blue stain or a lack of brown stain. If you've got no mitochondria, you can't do programmed cell death, apoptosis. And indeed, Laura went on to develop a mouse model, which we're still working with the aspirin trials. And what she found was if you knock out mitochondria, apoptosis goes down in a mouse that develops polyps. So we know that in green plants, salicylates, which is what aspirin's made from, drive programmed cell death to protect the plant against infection or to cause leaves to fall off in the autumn. And so programmed cell death is driven by salicylate in plants. It might be the same in humans. So it might be that as we get old, not only do the side effects go up, but the aspirin might lose its effectiveness. So we now recommend everyone stops taking aspirin from about 70 onwards, because the side effect profile gets really too big from that point on. The other half of the CAP2 and CAP1 trial was to look at a diet supplement. So this is Dennis Burkitt. And in fact, the whole idea of using aspirin came after the idea of using resistant starch or fermentable fiber. I happened to be introduced to Dennis by John Mathers, who was our professor of human nutrition in 1989, I think, at a seminar I was giving. And Dennis was very famous. He was a surgeon who worked in Africa, and he came back in the 1970s and said, you know, people in Africa don't get bowel cancer. And it's true that in rural Africa, colon cancer is really quite rare. And he suggested fiber was the reason. But actually, the Dunn Nutrition Unit he helped to found went on to demonstrate it was really fermentable fiber that was most important, what we now call in Britain resistant starch. And resistant starch looks like ordinary cornstarch, but it can't be digested in the upper GI tract. And so it reaches the bowel where it gets fermented into things like butyrate and short chain fatty acids, which are very good for the bowel. So we thought, well, we can randomize that too. So in our original trial, we randomized people to either get aspirin or placebo, or they got starch or placebo, or they got, you know, one or the other or both. And so if we just look at the starch limb now, The starch limb involved a total of 463 people who took starch compared to 455 who didn't. We gave them little packets to add to their food every day, either with cornstarch or with a resistant starch made by a company in America. It's called Novelos. It's used for making high fiber bread. And we knew that it might work on the bowel because, in fact, it didn't work in 2012. We analysed the data in 2012 and there was no effect of the starch. But we knew that it had worked in CAP1, not because it reduced the polyps particularly, but it reduced cell proliferation rate in the bowel. So we thought, let's go back and look at the data 20 years out to see whether that had an effect too. And to our amazement, it did, but not on bowel cancer. So the bowel cancer rate was exactly the same in both groups. But as a secondary analysis in the CAP2 trial, we said that we would look at all the other cancers, the non-colorectal cancers. And here the effect was really quite startling. And you can see from the graph there, these are the non-colorectal cancers. The people who were on the starch, who took per protocol, had dramatically fewer cancers in the rest of their body. And this was a planned secondary endpoint. So we had a lot of trouble getting this published because the referees kept saying, well, this is just data mining. But it wasn't. We said we were going to do this. And the effect was dramatic. So there was a more than 60% reduction in other cancers. This took us all by surprise. It eventually got published in 2022, and this won a prize as well from the American Cancer Group. And it was published by Cancer Prevention Research eventually. And we actually, the green bananas are because the best way of adding resistant starch to your diet is a slightly green banana, not too green, but just the ones you can still snap. Because the starch in a banana is initially crystalline and you can't digest it until it breaks down and turns sweet. So a sort of slightly firm banana gives you a lot of resistant starch. And so lots of my patients end up eating bananas. So why does it work? Well, the most dramatic effect was on the upper GI cancers. And we think it might be because changing the microbiome in the gut was affecting what are called secondary bile acids. So when you digest fat, your liver takes cholesterol and makes primary bile acids called deoxycholic acid and cholic acid, and they get put out into the bowel, through the duodenum, into the upper gut, and then they help you digest your fat, and then they go down into the bowel, in the large bowel, where they normally would get excreted. But if you've got the wrong kind of bacteria, they can be biotransformed into secondary bile acids. And these are nasty. They're carcinogenic and they get absorbed into the portal system, get sent back to the liver and then come out in the bile again. So they go round and round in circles. And they are basically potentially a driver of cancer in the duodenum and surrounding organs like the hepatobiliary tract itself. And we know in Lynch syndrome, these are places where we see more cancers. And when you actually, I haven't actually put the slide in, but basically we had 21 upper GI cancers in the placebo group and only 5 in the treatment group. So we think the biggest impact is on the upper GI tract. And the take-home message for Lynch patients is A, don't get overweight, and B, have a high fiber diet. So don't go overboard, because one of my patients, his wife phoned up and said, can you tell him to stop eating bananas? He's up to 23 a day and he's leaving the skins in bed. So, 1 or 2 bananas a day is fine. Moving on now to colon cancer endoscopy. This is always a bit of a worry for the colonoscopist, but basically there's 2 studies here that we've been involved in. One is the IMRC study, international study, looking at retrospectively at families where there was not surveillance to see what the cancer incidences were. And then our prospective database where all of the people were getting regular 1 to 3 yearly colonoscopy. And basically what was surprising was that with the exception of the green box, which is the PMS2 group, where there are clearly fewer bowel cancers in those on surveillance. If you look at MLH1 here, for example, if anything, there's slightly more colon cancers in the people who are on surveillance. And in fact, in Germany, where they do annual colonoscopy, they find significantly more cancers than in Finland, where they do 3 yearly colonoscopy. So we think what's happening is that the colonoscopy is not actually preventing cancer, because these cancers probably don't and usually don't have a dramatic adenoma stage, which you can resect. They do get what are called sessile adenomas, which are quite hard to spot. But we think that, in fact, a significant proportion of them just come out as cancer. Now, of course, it's still worth doing the colonoscopies, because these cancers, many of them will progress and they will eventually kill you if you don't take them out. So finding them is important, but we're not actually preventing the cancer with colonoscopy, as you might expect. And just to go back to our graph from our Aspen trial, the top line there, these are the people on the placebo group. And you can see that over the 20 years from 2000 to 2018, there was basically no difference in the number of cancers happening, despite them being on too early colonoscopy. So clearly there's something different about the biology in Lynch cases getting bowel cancer. Our partners in Germany have shed some light on this. They reported in 2012, Matthias Klur and colleagues, that about every square centimetre in the colon in these patients, you'll find, if you know how to look for it, a crypt which has lost its mismatch repair function, but is still working as a normal crypt. And these are the places where the spelling mistakes are going to accumulate. And we think that some of these just emerge as a cancer. And so Ursula Dover, who is the partner of Matthias Klur and ourselves, we partnered together with our biobank and published this paper in 2018, where we're suggesting that basically you can get to a cancer through the usual adenoma route, and that's particularly the case for PMS2. But for the other genes, you might frequently end up with a mismatch repair deficient crypt focus that then quite rapidly turns into a carcinoma. Or progressively, we might just get a mismatch repair deficient focus that just turns straight into a carcinoma. So you never get the chance to get ahead of it. Still important to detect it, but you can't get it before it turns to cancer. So Ursula and Matthias are brilliant scientists, and they've done some other amazing work. One of the things they looked at was metastases in these patients, which are quite rare. And when they looked at them, they found that the gene B2M, which has got one of these bits of repeat DNA in it, is frequently knocked out in cancers in the bowel, but in metastases, it was almost never missing. And so they think that that's because if you lose beta-2 microglobulin, which most of these cancers do, they essentially lose their HLA expression, and that makes them invisible. So I like to think of it like Harry Potter's invisibility cloak. So the tumors can grow in the bowel and not be visible to the local immune defenses. However, if they try to metastasize, they're traveling through the serum or the blood with no HLA on them. So the T killer cells will stop them. The state police stop them and say, "Can we see your passport, please?" They don't have one, so they get zapped. So we think that these tumors are likely to happen of their own volition, so to speak, and not prevented by adenoma resection. But the good news is they're slow to metastasize, so we've got plenty of time to find them. The other exciting bit of research that being led by Matthias and colleagues, which used our CAP bank, was to look at the mucosal biopsies in CAP2 and count how many T cells were present. And although it's a weak association, it's real, and it was published in Gastroenterology in 2022, that the people who have fewer lymphocytes in their colonic mucosa are more likely to go on and get a cancer. And so what we now think is that maybe some people are better at spotting these cancers than others, and that this is really a kind of immunological variation in the population, why some people can kill them and others can't. The other exciting development on the immunological side was the discovery of the immune checkpoint inhibitors. So this paper was published in 2005 at ASCO and led very rapidly to the license for the gene pembrolizumab as Keytruda in 2017, which is now soon to come off patent and is being widely used in oncology. And this is what's called a waterfall plot. The mismatch repair deficient colon cancers and mismatch repair deficient other cancers collapse in the presence of this drug, whereas those that are proficient don't. So what pembrolizumab does is it deregulates your immune system and allows it to kill anything. And it's sort of like telling the troops to go out and just shoot anybody they find. It's quite sort of damaging to your body generally, but it also massively effective against these cancers. So this is my simplistic cartoon. So your T cells are a bit like this guy, and they're wandering around, and they could kill anybody, but your cells all carry these inhibitors to say to them, "Please don't kill me, I'm on your side." So if you use these drugs, pembrolizumab and nivolumab are the PD-1 blocker, and the durvalumab, a more recent one, blocks the ligand itself, and then another one is ipilimumab, which blocks the CTLA4 receptor. So basically these drugs, and there's a whole host of them coming out, will deregulate your T cells because they block these inhibitors and allow it to kill cancers. And so increasingly, it's important to identify MSI high tumors because these are going to be the first line drugs. A very important paper about 18 months ago on rectal cancer showed that a bunch of people, about 13 of them, had rectal cancer treated with these drugs who were MSI high sporadic rectal cancers, and they went away without surgery. They completely killed them with the drug. So this is a very exciting new development. It means that finding these patients, whether they're sporadic or inherited, is increasingly important. And last few minutes. So how do we test for mismatch repair deficiency? Well, there's basically three ways of doing it. You can do immunohistochemistry for the protein. So this is one of Mark Aaron's pictures showing MSH2 missing in the tumor, the brown stain. You can use an old test called the Promega-Oncomate test, which uses very large fragments of repetitive DNA and looks to see whether they've gone wrong. But there's only 5 of them. And spotting these funny peaks against the stutter, as it's called, of normal PCR is quite hard to see. Or you can sequence and find the signature of MSI high in sequencing, but that's obviously expensive. So sequencing often fails because there isn't enough DNA. It's also expensive, and molecular pathology is already under stress. The workhorse solution doesn't work on next generation equipment, and mismatch repair testing using immunohistochemistry is relatively expensive. It costs about £250 per case if you look at the full cost. So we set out several years ago to invent a new test which would work on the next gen sequences, which uses little repeats, which are small enough to go through our short read sequences, and then pool them to give you an answer through a much cheaper, faster test. And just to prove why this is important, we just published this paper finally, which was led by Fiona McRonald and Stephen Hardy from the National Disease Registration Service, where we looked at 37,500 cancers in 37,000 people across England in 2019. Now, you'd expect about 32,000 of those to get to pathology. Not all of them do. And of those, you'd expect around about 1,000 new Lynch syndrome cases to turn up out of that group. What we in fact found was that 43% got MSI or IHC testing, but only 210 people got to a germline test. So that, we were missing hundreds and hundreds of Lynch patients because we simply weren't getting them through this very complex pathway. And so what and this is just to show in Northeastern Cumbria, we were only doing about 30%. Yorkshire was way ahead because Nick West and colleagues had got a grant to set up immunohistochemistry across the board. So it was an expensive approach, but they were testing about 70% of the cancers in Yorkshire. So we set up to develop this new test. And what we wanted to do was get a test that would run on the next-gen sequencer, but also include BRAF V600E and the driver RAS mutations. And the first bright idea from Mauro, who is a geneticist I work with here, was to look at the ones that had a linked polymorphism, like a blood group, that allows you to tell the one allele from the other. What that does is it takes away the noise, so you can differentiate a signal where it'll affect one allele from noise where it'll affect both similarly. So you can get the computer to chuck those away and clean up the message. And off the back of that, we got a grant with Kieran from our diagnostic lab to start rolling this out across the country. But we rapidly realized actually it was quite a hard test to do because it was using what's called molecular inversion probes. So while we were setting all this up in 5 of the 7 GLHs, we improved the assay dramatically thanks to the work Rachel did for her PhD, or was doing for her PhD. And this was because children who inherited Lynch syndrome from both parents get very early onset cancers. It's called constitutional mismatch repair deficiency. And we can actually pick that up on a blood test using our assay. But because we had DNA from these kids, we did whole genome sequencing to find the markers that were most likely to be unstable. And that developed a new set of markers that we could use in our assay. And so this is quite a complicated graph, but this is looking at the old score and the new score. So left to right is our old scoring system, where the difference between the stable and the unstable is quite small. Whereas using our new markers top to bottom, you can see that the stable cancers are dramatically different from the unstable cancers. And that is a very simple multiplex assay. This is what the person in the lab sees. So some wonderful software developed by Lizzie down in Sheffield. She's now at Northwest GLH. And Patricia, one of our master students, moved over to be a grade 6 tech. And so the grade 6 technologist sees this screen. The MSI result from the MySeq is shown here, which means that it's clearly MSI high. In fact, we don't say high and low anymore. The low thing is just an artifact of the old assay. So it's microsatellite unstable. There is BRAF present, and so this is a sporadic. The QC is fine. It doesn't need Lynch testing. And one technologist covers the whole region. So one technologist is now banging out 2,500 reports. And that's not on the colon cancers, but on the biopsies. So these are the biopsy blocks that come from the tumor. And we individually tested these because it was people were worried we might miss looking at the biopsies. And indeed, there are some biopsies that miss the cancer. But because we test the composite of all of them, we just take a slice across all the biopsies. This is a reliable assay. And so we can basically do this in a 6-day turnaround. And this is what the clinician sees. There's the test result, instability detected, clinical interpretation, in this case, increased likelihood of Lynch syndrome, and also that this patient has RAS mutations, is unlikely to respond to EGFR. So that report can be got out in 6 days by our lab now. It's about a 7-day turnaround. We're moving into weekend testing to try and get rid of the weekend delays. And as I say, one technologist can serve the whole Northeast and North Cumbria. And we're now, with support from Nick West and colleagues, rolling this out into West Yorkshire and beyond to try and make this much faster MSI BRAF testing, which will A, save money and time and expert resources, but also get more people tested for Lynch. We've just repurposed this TCAN robot from the COVID testing lab. So that goes live this month, which will allow us to do even more automated testing. And we're also adapting the assay we've adapted the assay to run on the NextSeq and are adapting it to run on the NovaSeq at the Marsden, so that you can use the assay alongside your other routine testing, and it becomes easier to put through. My last couple of slides, if I may. So we've already said the genes give different patterns of mutation, and particularly when it comes to the urothelial cancers. So urothelial cancers are the third most common cancer in Lynch syndrome. These are the yellow columns here. And if you've got MSH2, it's a quarter of people will get a cancer in their kidney or bladder. And around about 1 in 20 of all Lynch patients die of urothelial cancer. So Rebecca here, supervised by Rakesh, our urologist, and myself, and Gurdeep Sagu, is basically doing a study of can we pick up urological cancers with our assay. So at the moment, the European advice is that you should do blood testing in the urine and stuff, and basically it doesn't work. So this was a test done by the Mayo using the current recommended urine screening test. And they found 19 people out of 204 with hematuria, and none of them had a cancer. And there were 5 people who got cancer, and they missed them all. So that's 0% sensitivity and 0% specificity. Pretty useless test, which is why we don't do it. So we had a patient in CAP who had a renal cancer, Alan, down at Guy's, and we got his pre-op urine and we tested our markers, and sure enough, we picked up the abnormal markers from his tumour, which went away once he had his kidney removed. And there's one of those markers showing how very dramatically different it was. So we knew we could see his cancer. And then one of my patients, who was a 38-year-old scientist, had long-running menstrual bleeding, abnormal menstrual bleeding, and she'd been reassured by 2 gynecologists. But in fact, we got a tumour sample from her, and her strep urine sample and colibri urine sample clearly showed that she had cancer. She had a uterus out, and she had a high-grade mismatch repair deficient cancer requiring radiotherapy. The gynecologist had been misled by that fibroid, thinking that was the cause of her problems. So this urine test will pick up both both urinary tract and endometrial cancers. So we set up another study. We wrote out to all our MSH2 gene carriers. 83 of them sent us in samples, and very quickly these are the results. 76 of them were normal. One of them actually had developed bladder cancer while waiting for our clinic appointment, but he still had abnormal results after surgery, and that told them to go back in and have another go to get his bladder cancer out. This lady was a query false positive. She had a funny cyst. She had a slightly elevated urine test, which then went away spontaneously. We thought this thing in her kidney might be important, but it'd been there for 4 years, so it probably wasn't. So she maybe just naturally self-healed a urothelial cancer, which is why the urine test went negative. This chap already had a kidney cancer, and we found a large cancer in his other kidney, which they're going to try and get out without taking his kidney out. This lady had a 7.4 centimetre renal cell carcinoma, which actually isn't the sort of cancer we expected, but removing of that kidney took away the signal. This lady has 5 urothelial cancers in her kidneys and waiting for surgery. And this young man had a 10 millimetre cancer in his ureter, which was removed endoscopically. So basically it works. We also tested 18 patients just going through with urothelial cancers, and we picked up 2 patients, one with an MSH6 Lynch and the other an MSH2 Lynch. So we're now rolling this out to all our urothelial cancer teams, and hopefully that will go national. There's the lady with a colon cancer and a urothelial cancer. So this is now basically ready to roll out nationally. We put a grant application in to try and do this across the country, and we'll be hopefully starting that in the next few weeks. We're also going to try and add in some kidney cancer markers to make it an even more effective assay. These are the people who developed the urine test. These are the people who did the CAP trial. Haven't got time to talk about them all. So in summary, I should say we're going to have a conference in Barcelona if you want to come and talk about cancer and Lynch syndrome. So basically, these are the results. 4 key genes, 600 milligrams of aspirin, half your risk, resistant starch works. Old colons, maybe not so good. Metastasis is slow, but we might end up with a vaccine. Scopes don't prevent these cancers, but they catch them early. We've replaced IHC with MSI Plus, and we're hoping to use that as a urine screening test. And I will stop at that point, Nicola, and sorry if I went slightly over time.
No, you are perfectly timed. So thank you very, very much. Absolutely fascinating. And thank you for your time. I'm going to ask if there's any questions from the floor first. I've already had a couple of emails. You'll be pleased to know to say that they've thoroughly enjoyed the talk and it was absolutely fascinating. So thank you.
Lots of applause coming in. So there's nothing in the chat at the moment, John. So anybody who feels they just want to raise their hands or unmute. So we've got Adrian Hodson. You've got the hand up. You're on mute. Was it an accidental hand?
Sorry, accidental hand.
Yeah, no.
Oh, Adrian, you disappointed me.
It was a high five.
High five. Okay.
Anyone else?
One of the, just while we're waiting for anyone to put the hand up, I'll just use a few more minutes. I mean, getting the assay for MSI testing out is really quite a challenge, just the logistics of getting samples to the genomics lab. But what we're going to try and do is maybe get the Sheffield, well, Leeds are going to try and do the analysis of the data. So they'll ship us the samples to run through the robot, and then we'll send them the data and they can do the reporting. So we need to get around this sort of sample shipment issue, which is a general issue in genomics, of course, because the labs are so far away from the hospitals now. So Pascal put his hand up there, I think.
Yeah. Well done. Pascal, do you want to come in?
I'll take the slides down now because I think you've had long enough to read those.
Yeah. So Pascal, do you want to come in, or can you not unmute? I'm not sure. There is a comment in the chat. So it says, "Great talk, John. Fantastic work and nice to see it being largely Newcastle-led. Is there any therapeutic capability to mimic mismatch repair defects to artificially create vulnerability to immunotherapies in colorectal cancer, or is this likely to be too long in the production of mutant neoantigens?"
Oh, that's an interesting idea. Well, so first of all, obviously, you don't have to have mismatch repair to have a high tumor burden, a mutation burden. And so they're using immune checkpoint inhibitors more widely. But it's an interesting point. Can you kind of artificially knock down mismatch repair and then make the tumor more visible? I guess the problem always is, how do you get that just into the tumor and not into the rest of the body? Now you could imagine that working. So some of my friends have done like infusion treatments, where you put a catheter into the feed to the cancer. So you predominantly hit the cancer with those sorts of drugs, and Luke's nodding there as an expert in this sort of space. So, you know, you could imagine that working. And it's an interesting idea that you could potentially kind of make the cancer more visible. I think, first of all, we need to demonstrate in Lynch patients that we can get immune checkpoint inhibitors into routine use, because they're still being used as a backup treatment after they've hit them with the other stuff. And for example, 5FU doesn't work in mismatch repair deficient cancers. So we really need to get this MSI testing on all cancers. And that's one of the reasons we've pushed hard to get the automation running, so that it becomes a standard of care that all biopsies get tested for MSI. And Luke is nodding for those of you watching in black and white.
Yeah. So Pasquale's just come on as well. He's put his question in the chat because he couldn't get his mic to work, so he apologised, not to worry. And he said, I've done some work on MMR in prostate cancer. Usually 5% of met prostate cancer harbour defects, mainly sporadic in MMR. Could MSI plus work in prostate cancer too?
Yes, absolutely. So they're agnostic markers. We picked intergenic markers, so they will show MSI in any tissue. There's slightly lower levels of MSI in endometrial cancer caused by MSH6, because MSH6 is not a kind of frontline gene. It can be replaced probably by MSH3. So you don't get quite such a high level of mismatch repair deficiency with that gene. But with that exception, and it still works in endometrial cancer. In fact, we're going to be challenging NICE, who said we couldn't use it in EC because of old data using the old assays. Our assay is actually slightly better than immunohistochemistry in the Ohio series. So, but the general point is, yes, it should be used on prostate cancer. I mean, obviously I'm biased. I said at the start, you know, my name's on the patent, but you know, this test is there to drop in alongside everything else and just be used as a routine MSI test. So even if your whole genome sequencing stuff doesn't work, you'll still get your MSI result out. And it's just the logistics of getting sections cut. One of the things that I bang on about is that my pathology friends keep wanting to do tumor content. And I'm saying, since we're biopsying all the biopsies, we don't care what the tumor content is. And this assay will work down to 0.1 nanogram concentrations, which is like forensic levels. So it really isn't necessary to slow the process down to do tumor content analysis. You know, it's this sort of kind of making the process a bit slicker that tends to hold us up a lot, because the MDTs need this data when they see the patients, you know, when they discuss what to do about the patient. And increasingly, if you've got an MSI high tumor, especially in a patient, you know, who's maybe at risk of losing a big bit of their body, you might want to use immune checkpoint inhibitor first and see whether you can shrink or even destroy the cancer medically before you revert to surgery. So I think that could be the shape of things to come.
Great, thanks, John. And so we've got a question from Lisa Fisher, who's asked what your thoughts are on the inclusion of patients with Lynch into the bowel screening programs for colonoscopy and surveillance.
So thanks for that. Yeah. Kevin Monaghan, our partner in St Mark's, has led on this, and this is part of the transformation project. It was a good idea in the sense that it's giving the best quality colonoscopy to people with Lynch syndrome who are obviously in urgent need of this test. So when I said that it doesn't prevent cancer, it stops you dying of bowel cancer. And the thing is that colonoscopy is not a non-trivial intervention. It's actually as dangerous as aspirin, although people bang on about aspirin's risks and never mention colonoscopy. But you can die from colonoscopy too, especially if you're taking lumps off the ascending colon. And so putting these patients in the hands of the most expert colonoscopists and resourcing them better to do a good job is entirely defensible. It does mean the patients have to travel a bit further, but you know, I think that's defensible. And of course, what I'm hoping is we can add our urine test on alongside their colonoscopy, so we can screen them for urological and endometrial cancers. My real hope is that women in their late 30s, we might be able to use the urine test to be sufficiently reassuring that they can hang on to their uterus for a bit longer, because it's a real challenge for a woman over 35 with MLH1, say, who's faced with the prospect of hysterectomy to avoid cancer, but in the process losing the ability to have children. So I think, you know, that's our excitement, but we're waiting for the grant to sort of give us the resources to push that through. We submitted a bid to UKRI. We'll probably start rolling anyway, but we'll just have to do it a bit slower if we don't get the grant. as is usually the case.
Great. And then there's a lovely comment from Anne Paxton, who said that she was taught by you as a medical student in Newcastle more than 30 years ago. She's now in palliative care and comes across these patients and family members with inherited genes. So I have a great interest, though not directly involved with the genetics or the testing carriers. So yeah,
that's lovely to
really
hear.
nice.
Okay, Anne, can I just say, and I'm giving a talk to our palliative care doctors in a couple of months' time, because we've been saying for 30 years that we need to bank DNA from people who are dying.
Absolutely.
And actually, if it's presented to them as a sort of insurance policy for their family, so that if anything came along in the future that was of benefit to their descendants, the DNA is in the fridge and we can actually work out, you know, what the problem is. I understand why it's challenging to do, but I don't think that's a defense. I think palliative care should involve DNA banking as a standard. Because so often, and Helen will testify to this, you see people in the clinic, my auntie died of this, my uncle died of that, but did anybody do any tests? Nope. So we can't do tests on them without testing for everything. And, you know, that's just too hard. Whereas if you've got a DNA sample from the patient who died of the disease, then you've got something to compare to. So I think there's a real missed opportunity there, which I think I mean, it can even be done in a mouth swab now as well. It doesn't even need blood. Just to get something in the fridge for future use.
I completely agree with you, John. And I don't think families really appreciate the value of those samples until after the opportunity to gain them exists. So and I think also palliative care are also so skilled in having those discussions about kind of around death and what's happening and leaving relatives, that actually I think with matching those services, I think there's a real opportunity to introduce some of those conversations at an appropriate time. So yeah, I completely am on board with that. So we've got another question from Anne. Can you tell me when your talk is on the palliative care with the palliative care doctors, as she's still in the northeast? So if you want to.
So she will almost certainly know my dear friend Ellie Bond, who talked me into it after I'd been drinking wine. I think it's in about July, but I'm not absolutely sure. It's sometime before the summer.
So if you want to, I'll put my email address in the chat, and I can link you up with John, the secretary, so that you can make sure you find out when that is. And Carol Ann Hardy has also said, also patients and relatives ask a lot more about genetic risk and want to be involved. Excellent talk, thank you, and plenty of other thank yous coming in as well. It was a fantastic whistle stop tour, so thank you. Any more questions? Does anybody want to raise their hand and jump in, or pop another question?
Can I just raise two, just on that last point? That the PLSD, the Prospective Lynch Syndrome Database, it's www.plsd.eu. Will take you to that website. And if you see patients with this condition, it's really good to do that, because you can tell the website, it's written by my friend Paul Muller in Norway. You can say, this is a 42-year-old person, or 42-year-old man, what's his risk of prostate cancer? And he's got an MLH1 mutation, and it'll tell you, so it gives you patient-specific advice on their risk of each cancer, which is much better than just the general statements that we see in most of our sort of papers. And remember that a lot of the literature is based on retrospective analyses. These are all people who are being followed prospectively, so we know it's real. It's not sort of biased by ascertainment and so on. So I would urge you to use that. And the second thing is, if there's anybody on from Hull or Sheffield, we're really keen to get our MSI assay into you guys, and so we'd love to hear from you, because it's just the logistics. We're set up to do it. You just need to get the samples, and we can start, pairing up with more teams to do that.
Fantastic. Thanks, John. And fingers crossed there will be some further networking that answers that last question. I've also popped a link into the site that you mentioned, the Prospective Lynch Syndrome Database. I've popped a link into the chat there, so you should be able to access that after the meeting as well. So we're almost dead on 1 o'clock. So just goes to say many thanks for attending.